Welcome to the Deep Dive. Today we're tackling a truly fundamental mystery. How did complex life on Earth, actually begin?
Yeah, it's one of the biggest questions out there.
You've sent us a stack of fascinating sources looking into the origin story of eukaryotes.
Yeah.
That's us and, every other organism with complex cells, and the surprisingly central role of a group of microbes called Archaea. You might remember them as those weirdos that live in extreme places.
That's right. For a long time, archaea were seen as just a footnote in biology, maybe, those organisms thriving in boiling springs or incredibly salty environments, just oddities.
Right.
But what we've come to realize is that they represent a major branch on the tree of life, and in a truly astonishing twist, they're deeply connected to our own evolutionary history.
Okay, let's unpack this.
Yeah.
Because when I first heard Archaea, I thought, what do these extreme survivors have to do with complex life like us? That's the core of this deep dive, isn't it?
Absolutely.
To sort of extract from your sources this incredible link and show how the study of Archaea is completely reshaping our understanding of how complex life arose. And hopefully do it in a way that's engaging and reveal some truly unexpected connections without getting bogged down in too much jargon.
Precisely. We'll be tracing the ancient story of archaea, highlighting their unique characteristics, and some of them are quite bizarre, as you said, and then getting to their absolutely pivotal role in the evolution of eukaryotic cells.
The kind of cells that make up everything from a tiny yeast to an enormous blue whale.
And of course, you and me.
Yeah. So where does this epic story begin? It feels like we need to travel back like an immense amount of time.
Indeed. Our sources take us all the way back to LUCA.
LUCA.
The last universal common ancestor. Think of LUCA as the original ancestor of all life on Earth, living somewhere in the neighborhood of 3.6 to possibly even 4.3 billion years ago.
Wow, that's almost beyond comprehension. What kind of environment was LUCA inhabiting? Was it nice?
Probably not nice by our standards. The leading idea, supported by your sources, is that LUCA lived in deep-sea hydrothermal vents.
Okay. Dark, hot.
Picture this. Dark, oxygen-free environments, rich in minerals, hydrogen, and carbon dioxide, and likely very hot. LUC is thought to have been an anaerobic autotroph, meaning it didn't need oxygen and could produce its own energy using hydrogen and carbon dioxide, perhaps with some early forms of iron-sulfur chemistry. Simple, but effective.
So definitely not a tropical beach then. And there's even a clue that LUCA was a heat lover. That sounds interesting.
Yeah, what's fascinating here is the presence of a gene for an enzyme called reverse gyrase. This enzyme is commonly found in modern organisms that thrive in high temperatures, suggesting that LUCA itself might have been thermophilic, adapted to these hot conditions.
Okay, so LUCA is this ancient heat-loving microbe down in the deep sea, making a living from hydrogen and CO2. Pretty simple life.
Yeah.
What's the next big step in our story?
The first major split in the tree of life occurred around 3.2 to 3.5 billion years ago. The lineage that would become bacteria branched off from the lineage that would eventually lead to both archaea and eukarya.
That's a pretty fundamental divergence, right at the beginning almost. And where were the earliest archaea likely found? Still in those vents.
Probably in environments that were still anoxic and hot. Yeah, similar to LUCA's proposed home. This could explain why so many modern archaea are extremophiles, and it might be a holdover, from their ancient origins.
So from that early split, we eventually arrive at LSCA.
Right, LSCA, the last archaeal common ancestor.
Okay. What can you tell us about LSCA?
Well, LSCA is considered the ancestor of all the archaea we know of today. Based on what we can infer from their modern descendants, LSCA was also likely hyperthermophilic, really flourishing in intense heat.
Still loving the heat.
Seems so.
Yeah.
And a key metabolic innovation that emerged within the archaeal line, although perhaps not in LSCA itself, but very early within one major group, the Eukaryotic, is methanogenesis, the production of methane. We actually have geological evidence suggesting methane-producing archaea existed as far back as 3.46 billion years ago.
Methane production. That's certainly a unique talent. So LACA itself might not have been a methane producer. That's an interesting nuance.
It is. Some research indicates that methanogenesis developed slightly later within a specific branch of archaea. And what makes it even more complex is the possibility of horizontal gene transfer.
Ah, gene swapping.
Exactly. The swapping of genetic material between organisms which could have spread the ability to produce methane to different groups of archaea.
So it's not a completely clean family tree. There was some sharing going on.
Absolutely. Early life was likely a period of significant genetic exchange, lots of mixing and matching.
Okay.
But beyond these specific metabolic abilities, what's truly remarkable is that LACA likely possessed the early forms, the sort of precursors to the complex information processing systems we see in eukaryotes today.
You mean like DNA and stuff?
Yeah, the machinery for copying DNA, reading it to make RNA, and then using that RNA to build proteins. This suggests a level of sophistication in this ancient archaeal ancestor that might come as a surprise.
It already is surprising me. And it looks like we have a helpful timeline in Table 1 of the sources to keep all these billions of years straight.
Yeah, that table's useful. It lays out the origin of water, LUCA, that big bacterial archaeal split, the first hint of methanogenesis, LACA, the GOE, we'll get to that, and then finally, eukaryotes.
It really provides a sense of the immense timescales involved. Mind-boggling, really.
It does. It underscores the vast amount of time over which these fundamental evolutionary steps unfolded. So now let's focus on what makes Archaea distinctly Archaea.
Right.
They are prokaryotes, like bacteria, meaning they lack a nucleus and other complex internal compartments.
Okay, basic biology. Got it.
But that's where many of the similarities start to fade.
Right. I recall that much. But the sources highlight some truly distinctive features, starting with their cell membranes. Let's dive into these unique structures.
This is a critical distinction, a really fundamental one. Bacteria and eukaryotes have membranes made of fatty acids linked to a molecule called glycerol-3-phosphate via ester bonds. Standard stuff.
Okay.
Archaea, on the other hand, have isoprenoid chains, different building blocks linked to glycerol-1-phosphate through ether linkages, different chemistry.
Isoprenoids. Ether linkages.
Sounds technical. Why is this lipid divide so important?
Well, the seemingly minor chemical difference had profound implications. It essentially gave Archaea the molecular armor to conquer environments where other life couldn't easily survive.
So the ether linkages are tougher.
Exactly. The ether bonds in archaeal membranes are chemically much more stable, especially when faced with high temperatures and extreme acidity or alkalinity. This is a major reason why many Archaea can thrive in these harsh environments.
Makes sense. Built for resilience.
And some Archaea even have a tetraether lipid monolayer, where the isoprenoid chains stretch across the entire membrane, creating exceptional rigidity. Imagine a super reinforced cell boundary. Wow. Okay, so their membranes are built tough.
Right.
What about their cell walls? Bacteria use peptidoglycan, correct? I remember that one.
Correct. But Archaea don't have peptidoglycan. Instead, their cell walls show remarkable diversity. It's not one size fits all.
So what do they use?
Some methane-producing Archaea have something called pseudomurane, which is structurally similar to peptidoglycan, but built from different components, sort of an imitation.
Okay.
Many other Archaea possess S layers, which are intricate arrays of proteins or glycoproteins forming the outermost layer, providing structural integrity and protection, like chainmail made of protein.
So a real evolutionary flexibility in how Archaea construct their outer defenses.
Exactly. You see real variety there.
And they even have a unique way of moving around, not the bacterial flagellum, I gather.
Precisely. Many Archaea use a structure called the archillum for motility.
Archaeum.
While it serves a similar function to the bacterial flagellum, it rotates to propel the cell. Its underlying structure and the way it's powered are completely different. It looks similar on the outside, maybe, but it's built differently.
How so?
The archaeolum is actually more closely related to bacterial-type IV pili, structures often used for attachment or twitching motility, and it's driven by ATP energy, not the proton flow that powers bacterial flagella.
Okay.
It's another fascinating example of convergent evolution, where a similar function, swimming, evolved through distinct evolutionary pathways.
Okay, so unique membranes, diverse walls, and a different kind of propeller.
Yeah.
But the sources also highlighted some surprising similarities between Archaea and Eukarya, especially when it comes to their genetic systems.
Yes.
This is where the connection to us really starts to solidify, right?
Absolutely. This is crucial. When we examine the machinery for DNA replication, copying DNA,
Right.
transcription reading DNA to make RNA, and translation using RNA to build proteins, in Archaea, we see much closer parallels to eukaryotes, to us, than we do to bacteria.
Give us some specific examples in DNA replication. How similar are we talking?
Well, okay, take the very start of replication. Archaea possess homologs, similar versions of key eukaryotic proteins involved in initiating DNA replication, like ORC1-CDC6, which identifies where replication should begin.
Your starting signal.
Exactly. Their DNA unwinding enzyme, the helicase, is the MCM complex, just like in eukaryotes. They also use pCNA as a sliding clamp to help the DNA copying enzyme work efficiently.
Okay.
Again, like eukaryotes, but different from the bacterial beta clamp. Even the enzyme that lays down the initial RNA primers to start DNA synthesis, called primase, is a two-part protein, prispryl, in both Archaea and Eukarya, unlike the single-part DNAG primus in bacteria.
Wow. So the starters, the unwinders, the clamps, the primers, lots of similarities.
It's a remarkable number of detailed similarities in such a fundamental process, yeah.
What about transcription, the step where DNA is transcribed into RNA? Same story.
The similarities continue. Archaea use a single RNA polymerase, the enzyme that performs transcription,
Right.
which, in its structure and how it functions, bears a greater resemblance to the eukaryotic RNA polymerases. We have three main ones, but they're related then to the bacterial one.
Ah.
They also employ general transcription factors such as TBP, tata-binding protein, and TFB, which is related to the eukaryotic TFIIB, to initiate transcription at gene promoters, just like in eukaryotes. Some even have a homologue of TFIIE.
These are like the key managers for starting gene reading in our cells.
Exactly. These are crucial components of the eukaryotic transcription initiation complex, and Archaea have versions of them.
And translation, building the proteins, don't tell me.
Once again, the archaeal machinery shows a closer affinity to eukaryotes. Their ribosomes, while similar in overall size to bacterial ribosomes, 70S, contain more ribosomal proteins, and many of these are homologous to eukaryotic ribosomal proteins.
The more complex ribosomes.
Structurally, yeah, closer to ours. And key factors that start the process of translation, like AIF1 and AIF2, are direct counterparts of eukaryotic EIF1 and EIF2. The elongation factors, which help add amino acids to the growing protein chain, also exhibit greater similarity to their eukaryotic counterparts.
This is really compelling evidence for a closer evolutionary relationship between Archaea and Eukarya. It's not just superficial.
Not at all. It's deep in the core machinery of the cell.
And there's even mention of histone-like proteins in Archaea, like what we use to pack our DNA.
Yes. Many Archaea have histone proteins that can compact their DNA, much like the core histones in eukaryotic nucleosomes wrap up our DNA.
So they were already organizing their DNA.
It suggests that the fundamental mechanisms for organizing genetic material might have evolved early in the archaeal lineage, providing a foundation for the more elaborate chromatin structures found in eukaryotes. It's as if Archaea possess some of the basic building blocks of eukaryotic complexity within their own simpler cells.
That's amazing. And Table 2 in your sources kind of lays all this out, right?
Yeah.
Comparing the three domains.
Yeah, Table 2 provides a really clear summary of these key differences and similarities across various features, cell walls, membranes, genetics, information processing, all that stuff for Archaea, bacteria, and Eukarya. It's a fantastic visual overview.
It is. It really emphasizes that Archaea are not just unusual bacteria. They possess their own distinct identity and, wow, a surprising number of connections to us. Now, we've discussed their unique features in these shared genetic systems. What about their metabolism? We know they inhabit extreme environments, but what else are they capable of? Is it all just heat and acid?
No, not at all. While their reputation for thriving in extreme conditions is well-deserved, some live at incredibly high temperatures or very low pH. The metabolic diversity within Archaea is actually remarkably broad. They're not just specialists.
Okay.
They play crucial roles in global carbon, nitrogen, and sulfur cycles, even in more modern environments like oceans and soils. They're everywhere, doing important jobs.
So they're not just living on the fringes. They're vital for the planet's overall health, like globally important.
Absolutely. And they have some unique metabolic tricks up their sleeves. Methanogenesis, as we discussed, is exclusive to certain Eukaryotes. Only they can do that.
Right, the methane makers.
Others can oxidize ammonia. The Eukaryotes are really important for that in the oceans. Others use sulfur or iron. They can be chemolysotrophs, getting energy from rocks, essentially.
Wow.
Or chemoorganotrophs, utilizing organic compounds like we do. Some can fix carbon dioxide like plants, while others need ready-made carbon. Huge diversity.
And the Asgard superphylum, which keeps coming up in connection with eukaryotes.
Yeah.
What's their metabolic story? Are they specialized?
The Asgard Archaea actually exhibit significant metabolic flexibility. Genomic analysis suggests they can break down a range of organic compounds, use a specific pathway called Wood-Dahl for carbon fixation, and engage in hydrogen metabolism, both producing and consuming it.
Hydrogen seems important here.
Very. Some Asgard lineages can even ferment organic matter or, surprisingly, perform aerobic respiration or use nitrate instead of oxygen.
Wait, aerobic respiration? Like us?
Potentially, yes, though likely through genes acquired from bacteria via that horizontal gene transfer we talked about. It highlights their adaptability.
So they can borrow tricks from bacteria too.
It seems so. And this metabolic versatility, especially the hydrogen connection, would have been critical for establishing the kinds of symbiotic relationships, the partnerships, thought to have led to eukaryotes.
Which leads us perfectly to how these seemingly simple archaea are actually central to the origin of complex life eukaryogenesis. This is the big aha moment, isn't it?
It is indeed. Eukaryogenesis was a truly transformative event in the history of life. It resulted in the compartmentalized cells with a nucleus and organelles that define all eukaryotic life, from fungi to plants to animals.
And the key theory explaining this is endosymbiosis, but now with a major archaeal twist.
Exactly. The spotlight is firmly on Archaea now.
So remind us of the core principles of endosymbiotic theory, just briefly.
Essentially, it proposes that key eukaryotic organelles originated from symbiotic relationships where one prokaryotic cell engulfed another and they started living together. The most pivotal event, the one that likely kicked things off, was the engulfment of an alpha proteobacterium.
A type of bacteria.
Right, which eventually evolved into the mitochondrion, the powerhouse of the eukaryotic cell responsible for aerobic respiration.
And there's strong evidence supporting this bacterial origin of mitochondria, right? It's not just a guess.
Oh, absolutely. The evidence is compelling. Mitochondria have their own circular DNA, similar to bacterial chromosomes, though much smaller now. They possess double membranes, with the inner one resembling a bacterial membrane.
Like the original bacterium's membrane.
Precisely. They use bacterial-type ribosomes and even reproduce by binary fission, sort of like bacteria dividing independently of the host cell cycle.
And genes moved.
Yes. Over time, many of the genes originally in the mitochondrial DNA move to the host nucleus, solidifying their interdependence, making it a permanent arrangement.
Right. And chloroplasts in plants, same idea.
Same principle, yes. Chloroplasts, found in plants and algae, arose from a later, separate endosymbiotic event involving a cyanobacterium, a photosynthesizing bacterium.
Okay, so these endosymbiotic events weren't just about acquiring an organelle. They fundamentally reshaped the host cell's architecture and, crucially, its energy production.
They really did. It was a merger that changed everything.
So a bacterium gets swallowed, becomes a vital organelle. But who was doing the swallowing? Who was the host cell? For years, that was a mystery. But this is where the Asgard Archaea come into play, right?
Exactly. For a long time, the identity of the engulfing host was a major puzzle. Was it an early eukaryote that somehow lacked mitochondria? Was it some other kind of prokaryote? But the discovery of the Asgard superphylum of Archaea has provided the most compelling answer yet.
How were these Asgard Archaea discovered? Their names sound like something straight out of Norse mythology. Loki, Thor.
They were. Their names Loki Archaeota, Thor Archaeota, Odin Archaeota, Heimdall Archaeota are indeed inspired by Norse mythology, reflecting their discovery in sediments near a hydrothermal vent field called Loki's Castle in the Arctic Ocean initially.
Cool names. But how did we find them?
They were discovered not by growing them in a lab initially, that's incredibly difficult, but through metagenomic sequencing. Scientists were analyzing DNA samples taken directly from diverse, often oxygen-deprived environments like deep-sea sediments.
Sifting through environmental DNA.
Exactly. And what the genomic data revealed was astonishing. These archaea are phylogenetically, in terms of their evolutionary relatedness, the closest known prokaryotic relatives of eukaryotes.
Closer than any other archaea. Or
Yeah.
bacteria.
By far. Some studies even suggest that eukaryotes didn't just arise alongside Asgards as a sister group, but actually emerged from within this archaeal group.
Wait, hang on. So we didn't just split off near them, we came out of them.
That's what the latest phylogenetic trees strongly suggest. Specifically, a group within Asgard called Heimdall Archaea, and maybe even more specifically, Hodarchaeals within that, often comes out as the closest relative branch.
That's a real game changer for our understanding of the tree of life. So instead of 3 distinct domains, bacteria, archaea, and eukarya.
We might be looking at just 2 primary domains, bacteria and archaea, with us eukaryotes being a specialized, very fancy branch that budded off from within the archaea, specifically the Asgards.
Wow, the textbooks need updating.
This view, often called the 2-domain tree, strongly supports an older idea called the eocyte hypothesis, and really emphasizes the deep archaeal roots of eukaryotic complexity. The term eukarymorpha has even been proposed to describe this Asgard eukarya clade, highlighting their shared features in history.
Eukaryomorpha. Okay. And the big piece of evidence linking Asgards to eukaryotes, beyond just the overall DNA similarity, is the presence of those eukaryote signature proteins, or ESPs. Tell us more about those. What are they?
This is where things get truly fascinating, really mind-blowing. Asgard Archaea possessed genes for proteins that were once thought to be exclusively found in eukaryotes' hallmarks of eukaryotic complexity.
Like what? Give me examples.
Proteins involved in things like building the cell's internal skeleton, the cytoskeleton, things like actin. Proteins involved in moving things around inside the cell in vesicles, which is called membrane trafficking. Components of the system for tagging and degrading proteins, the ubiquitin system. And even proteins involved in complex information processing and regulation.
So like the fundamental machinery that makes our cells complex.
Exactly. Think of these ESPs as the foundational tools, the basic toolkit that underpin eukaryotic complexity. And Asgards have them, or at least proto-versions of them.
So these Asgard Archaea had the genetic instructions for some of our complex cellular machinery even before they encountered the proto-mitochondrion. That seems backwards.
That's the remarkable discovery. It implies the archaeal host wasn't just a simple passive recipient. These ESPs and Asgards are likely earlier, perhaps simpler versions of their eukaryotic counterparts. They might have performed more basic functions in the archaeal cell.
Like what? Why would they need them?
Good question. The evolutionary pressures that led to these ESPs and Asgards before endosymbiosis aren't fully understood, but they might have been related to things like enabling interactions with other cells, perhaps in those metabolic partnerships we talked about.
Ah, for syntrophy.
Possibly. Or maybe for maintaining a specific cell shape, interacting with the environment, or handling internal organization. Table 3 in your sources gives a great overview of some key ESPs found in Asgards and their potential roles. For example, they have actin-like proteins, like Loki actin, that could have been involved in shaping the cell and forming protrusions.
Protrusions, like little arms.
Sort of, yeah. They also have components of the ESCRT complex and snare proteins, which are crucial for remodeling membranes in eukaryotes. Think vesicle formation, cell division.
Stuff we use all the time.
Right. And even parts of the ubiquitin system, which in us is vital for regulating almost everything, but in them might have had simpler roles in stress response or protein quality control.
This paints a picture of Asgard Archaea as being surprisingly sophisticated, or at least having the potential for complexity even before they became the ancestors of eukaryotes. And are we actually seeing evidence of this complexity in real Asgard cells, since they're so hard to grow?
Exactly. While growing Asgard Archaea in the lab has been incredibly challenging, there have been some recent breakthroughs, and techniques that allow us to study their genomes and even visualize them in their natural environment or in co-culture have revealed that some of them have quite intricate structures.
Like those protrusions you mentioned.
Yes, some cultivated ones like Candidatus prometheoarchaeum centrophicum show these long branching extensions reaching out from the main cell body. And advanced microscopy on another one, Calotlochiarchaeum ossiferum, has even shown an actin-based cytoskeleton that seems to support these extensions.
So they really do have a kind of internal scaffolding.
It seems so. While they don't have membrane-bound organelles like eukaryotes, this level of internal organization and the clear potential for membrane remodeling, based on those ESPs they have, suggest a degree of complexity we really didn't anticipate in prokaryotes.
Not just simple blobs, then.
Definitely not. Some studies looking at Hodarchaea's genomes even suggest they might have larger cell sizes and perhaps a more organized arrangement of their genetic material within the cell, maybe precursors to a nucleus, though that's still speculative.
And this pre-existing complexity in the Asgard host, that could have been crucial for the whole endosymbiosis thing, right? For interacting with the bacterium.
Absolutely. It could have been crucial for facilitating the initial interaction with and eventual engulfment or trapping of the protomitochondrion. Maybe those protrusions helped, even if it wasn't exactly the same kind of engulfment phagocytosis that we see in amoebas today. This inherent complexity likely set the stage.
Okay, so with this updated picture of a complex Asgard archaeon, how do the different models of eukaryogenesis stack up now? It sounds like they all have to involve an Asgard-like host now, right? That's non-negotiable.
That's largely the consensus, yes. Most current models now place an Asgard archaeon at the center of eukaryogenesis. The main differences lie in how they propose the initial relationship with the alpha proteobacterial ancestor of mitochondria actually began. What was the first step? Table 4 in your sources provides a helpful summary of these different models and how Asgard discoveries have influenced them.
Let's touch on a couple of the main ones. Syntropy-based models seem popular, like the hydrogen hypothesis. What's that?
Yes, syntrophy-based models propose that eukaryogenesis started with a metabolic partnership, a dependency. The hydrogen hypothesis, a classic example, suggests a relationship where a bacterium producing hydrogen, the proto-mitochondrion, and an archaeon consuming hydrogen, an Asgard-like organism, lived in very close association, basically needing each other's waste products.
Okay, metabolic marriage of convenience.
Pretty much. The reverse flow model is similar, but proposes the opposite flow. Maybe an Asgard archaeon producing hydrogen or electrons that the alpha proteobacterium consumed.
Either way, they're swapping chemicals.
Exactly. And the metabolic versatility we observe in Asgards today, their ability to use hydrogen and break down organic matter, really supports the idea that a metabolic exchange could have been the initial driving force pulling these two microbes together. And those ESPs involved in cell, shape, and interaction would be really important for maintaining such a close physical partnership.
Right, those protrusions maybe help them stick together or exchange things. What of the idea of a phagocytosing archaeon? Did the Asgard host just, eat the bacterium?
The phagocytosing archaeon model suggests that the Asgard host already had some capacity for engulfment, perhaps rudimentary, facilitated by its early cytoskeleton, like actin, and membrane remodeling machinery, those ESCRT components, snares.
Using the ESPs it already had.
Precisely. While we don't observe fully developed phagocytosis in the Asgards we've been able to study so far, those membrane extensions they possess could represent an early step in that direction, or perhaps a different, simpler mechanism of uptake was involved initially. It's still an active area of debate whether full phagocytosis came first or evolved later, maybe after the initial symbiosis was established.
So maybe they teamed up metabolically first, got really close, and then one engulfed the other.
That's a very plausible sequence of events favored by many syntrophy models.
We should briefly mention some of the other ideas, like viral eukaryogenesis or the inside-out model.
Yeah.
Are they still in the running?
They're less mainstream now, perhaps, but worth noting. Briefly, the viral eukaryogenesis hypothesis proposes that the eukaryotic nucleus might have originated from a large virus that infected an archaeon. The inside-out model suggests that the cytoplasm and the internal membrane system of eukaryotes arose from outward protrusions or blebs of an ancestral archaeal cell, with the original cell body becoming the nucleus that then engulfed the protomitochondria living on its surface.
Interesting ideas.
While they offer alternative explanations for certain eukaryotic features, like the nucleus or the complex internal membranes, they generally still involve an archaeal host, and Asgards remain the best candidate for that host.
Okay, so regardless of the precise mechanism, whether it was partnership first or engulfment first, it seems increasingly clear that an Asgard-like archaeon was the cradle, the host, in which the first eukaryotic cell arose.
That's the picture emerging, yes.
So what about LCA, the end result of this merger? The last eukaryotic common ancestor. What did it look like? Was it simple?
Surprisingly, no. Based on comparing the genomes of all living eukaryotes from protists to fungi to us, LCA was already remarkably complex, far more complex than any prokaryote.
Really? What did it have?
Reconstructions suggest LCA already had a nucleus containing linear chromosomes, mitochondria for energy, an endomembrane system like the ER and Golgi, a dynamic cytoskeleton, interruptions in its genes called introns, and sophisticated ways to regulate gene activity. It was, in many ways, a modern eukaryotic cell in blueprint.
Wow. So a lot of complexity emerged very early. And its genome was a mix.
Absolutely. Its genome was a fascinating mix, a true mosaic. It contained genes originating from the archaeal host, genes from the bacterial endosymbiont, the mitochondrion, and even genes acquired from other bacteria through horizontal gene transfer over time, a real chimera.
So, at the very deepest level, we are a true hybrid organism. What were the main contributions from the archaeal side to LECA? What parts of us are fundamentally archaeal?
The archaeal ancestor, very likely in Asgard, contributed significantly, massively, to LECA's core information processing systems.
The genetic stuff again?
Yes. The machinery for replicating DNA, transcribing it into RNA, and then translating that RNA into proteins, the core components, the key proteins involved like ORC1, CDC6, MCM, PCNA, TBP, TFB, RNA pol subunits, many initiation and elongation factors for translation largely have archaeal origins.
So the whole operating system, basically.
You could think of it that way. They also provided the fundamental building blocks for the cytoskeleton, like actin and tubulin. The endomembrane system involved in trafficking, including components like ESCRTs, snares, RAB GTpases, the machinery for transporting molecules in and out of the nucleus, pathways for sorting proteins within the cell, and the ubiquitin system for protein regulation and degradation.
So the information systems and the basic cell structure and organization.
A huge chunk of it, yes. Plus some metabolic contributions too, like aspects of glycosylation, adding sugars to proteins, making certain lipids, some amino acid synthesis, and DNA repair pathways. Essentially, the archaeal host provided the foundational framework and operating system of the eukaryotic cell.
And the bacterial contributions, primarily the mitochondrion for energy production, I assume.
Exactly. The alpha proteobacterial endosymbiont that became the mitochondrion was arguably the most significant single bacterial contribution because it revolutionized energy metabolism. It provided LEA with the ability for highly efficient aerobic respiration using oxygen.
The powerhouse.
The powerhouse, exactly. Genes related to mitochondrial function, such as those involved in the electron transport chain, oxidative phosphorylation, the TCA cycle, making iron-sulfur clusters, these are predominantly of bacterial origin. The acquisition of high-energy molecules, high redox potential ketones, crucial for aerobic respiration, also came from this endosymbiont.
Any other bacterial bits?
Beyond the mitochondrion, some other bacterial genes were likely acquired through horizontal gene transfer along the way, contributing additional metabolic capabilities, maybe enzymes for anaerobic pathways, or breaking down specific compounds.
So this early eukaryotic cell, Archaea, was a remarkable combination of archaeal structure and information processing and bacterial energy generation. But what environmental factors then shaped its evolution and diversification? The Great Oxidation Event must have played a major role, right? All that new oxygen.
Absolutely. The Great Oxidation Event, the GOE, which really took off around 2.45 to 2.2 billion years ago, marked a significant permanent increase in the amount of oxygen in Earth's atmosphere and oceans, thanks to cyanobacteria doing photosynthesis.
Changed the whole planet.
Fundamentally, this had a profound impact on the evolution of life, including these early eukaryotes. While Archaea and early eukaryotes likely evolved in environments with low or fluctuating oxygen levels, they probably still had ways to live anaerobically.
Okay.
The GOE created a strong selective pressure favoring the refinement and widespread use of aerobic respiration, which the newly acquired mitochondrion provided. Oxygen was initially toxic, but mitochondria offered a way to not just detoxify it, but use it.
A huge advantage.
Massive. The increased energy yield, the ATP, from aerobic respiration likely fueled the evolution of larger cell sizes, greater complexity, multicellularity, and the subsequent diversification of eukaryotes into the vast array of forms we see today. Oxygen provided the fuel for complexity.
But there are hints they weren't fully aerobic initially.
Yes, the fact that many modern eukaryotes, even complex animals, still retain anaerobic metabolic pathways, and that some core mitochondrial enzymes seem to function optimally at low oxygen levels, hints at their origins in a less oxygenated world. Archaea was probably facultatively anaerobic. It could live with or without oxygen.
Flexible. And beyond this massive environmental shift with oxygen, were there other genetic influences at play, like ongoing horizontal gene transfer? Did eukaryotes keep borrowing genes?
Yes, HGT hasn't stopped. It has continued to be a factor in eukaryotic evolution, particularly in single-celled eukaryotes, the protists.
By especially them.
Well, many protists actively engulf other cells for food phagocytosis, which naturally increases the opportunities for DNA from the prey to get mixed in and potentially incorporated into the protist's genome.
Accidental gene uptake during lunch.
Something like that. So HGT allows eukaryotes, especially protists, to acquire new genes and metabolic capabilities from bacteria, archaea, even other eukaryotes, aiding in their adaptation to new environments, new food sources, new lifestyles.
Like getting metabolic shortcuts.
Exactly. There's also this concept called the HGT ratchet. If a gene gets transferred from an organelle like a mitochondrion, or even a temporary symbiont, to the nucleus, the original copy might be lost from the organelle. This tends to accumulate foreign genes in the nucleus over time, further contributing to that mosaic nature of eukaryotic genomes we talked about.
So it's a one-way street for some genes, locking them into the nuclear genome.
Often, yes. Examples include acquiring enzymes for anaerobic metabolism in eukaryotes living in oxygen-poor environments, or pathways to break down unusual compounds. It's a continuous source of evolutionary innovation.
It's incredible to consider this constant genetic give and take shaping life over such immense timescales. Now, as we begin to wrap up this deep dive, what are some of the major unanswered questions in this field? What are scientists still trying to figure out about the origins of Archaea and their role in our own beginnings? Where's the edge of knowledge?
Oh, there are still several really exciting areas of active research and debate. One big one is pinning down the precise evolutionary relationships within the deepest branches of the Archaea and the exact placement of the Asgard superphylum within that tree.
So the family tree is still a bit blurry in the deep past.
It is. Different studies using different gene sets or different computational models can sometimes yield slightly different results about whether eukaryotes are sister to all Asgards or emerge from within a specific lineage like Heimdall-Archaea hodarculars. Things like long branch attraction artifacts in tree building or compositional biases in DNA or genes getting lost differently in different lineages make it really challenging to get a definitive, stable picture that far back. New data and better methods are crucial.
Okay, refining the tree. What else?
Another key question is understanding the selective pressures and the evolutionary steps behind those eukaryote signature proteins, the ESPs. Why did they arise in Asgard Archaea before the mitochondrial endosymbiosis?
Yeah, that's the big puzzle. What were they doing?
Exactly. What were their original functions within those archaeal cells? Was it for centrophy, cell shape, something else? And then how did these simpler proto-ESPs evolve and become elaborated into the much more complex and integrated systems we see in eukaryotes? How did gene duplication, shuffling bits of proteins around, gaining new functions, neofunctionalization play out? And what role did the arrival of the mitochondrion play in driving this elaboration? That interaction is key.
How the partnership itself changed the host's toolkit.
Right. And finally, the lipid divide, that fundamental difference in membrane lipids between Archaea, ether-linked isoprenoids, and bacteriocharya, ester-linked fatty acids, remains a significant puzzle, a huge one.
How did that swap happen? It seems like changing your entire skin.
It's a major biochemical hurdle. How did the transition from an archaeal host membrane system to the bacterial eukaryotic type occur during eukaryogenesis? Was it a massive transfer of bacterial genes for making fatty acids? Did they have chimeric membranes for a while, a mix? Did viruses play a role, or was it a gradual replacement?
And the centrophy models might be relevant there too, if they were physically close.
Exactly. Close physical interaction might have facilitated lipid transfer or gene transfer for lipid synthesis pathways. Finding the answer is crucial for understanding the very foundation of the eukaryotic cell membrane.
So, even with all the incredible progress we've made, identifying Asgards, finding ESPs, there are still fundamental mysteries to unravel about our deepest origins.
Absolutely. It's a very active field.
But the central role of Archaea in this story is now, it seems, firmly established. They're far more than just odd extremophiles living in weird places.
Much, much more. They're key players in the very genesis of eukaryotic complexity.
Right, so the big takeaways are eukaryotic cells, our cells, are truly chimeras, hybrids.
Precisely. Born from the merging of an archaeal host, most likely from the Asgard lineage, and a bacterial endosymbiont that became the mitochondrion.
And the archaeal contribution was immense. The core informational systems, the genetics, the translation, and the basic building blocks of our cellular architecture, the cytoskeleton, the internal membranes.
The framework and the operating system, yes.
Well, the bacteria, primarily through the mitochondrion, provided the revolutionary power source of aerobic respiration.
Harnessing oxygen, yeah. A critical innovation.
And it wasn't instant. It was a long, intricate evolutionary journey involving coevolution, partnership, gene transfer, repurposing existing tools, exaptation, and the gradual emergence of the features that define us as eukaryotes.
A protracted process, absolutely. And while mysteries still exist, the fine details of the phylogeny, the full story of ESP evolution, that stubborn lipid divide, the ongoing study of Archaea, these incredible microbes, continues to illuminate this amazing story of our own origins.
It truly makes you contemplate. This seemingly simple microbial world, often invisible to us, holds the key to understanding the very fabric of complex life, including ourselves. It really makes you wonder, what other fundamental secrets might still be hidden within these ancient lineages, waiting to be discovered?
A profound thought.
Something for you to mull over. Thanks for taking this deep dive with us today.